I'm equally thrilled to introduce our second speaker today, Dave Micklos. Dave studied biology at Salisbury University in Maryland, and then journalism at the University of Maryland. He joined Cold Spring Harbor Lab as the Director of Public Affairs and Development, but soon after founded and then became the Executive Director of Cold Spring Harbor Lab's famed DNA Learning Center. This is a hands-on learning initiative, now programs for teaching DNA and life sciences for middle school and high school kids, as well as teachers and the public at large. And hopefully you'll hear a lot about that today. They run several centers directly in the area and the establishment of several centers in the country and around the world with enormous impact. Their kits go out to about 500,000 kids each year, and their internet portal attracts over 5 million views. Dave authored 3 textbooks, received the Dana Award and the Genetic Society Award for Excellence in Education, the Science Prize for Online Resources in Education, among other things. He's a fellow of the AAAS as well. I wanted to add that the first time I encountered Dave shortly after coming to Cold Spring Harbor Lab a few years ago at this point, he gave a fascinating talk about eugenics in the early part of the 20th century in the United States. It was an astounding and brilliant talk. And as you can see, it left a lasting impression on me.
Well, I appreciate the opportunity to talk to colleagues near and far about our work at the DNA Learning Center of Cold Spring Harbor Laboratory. Our site has been a place of education since the mid-1800s, when there was an elementary school on this site, and then we took over the current building and renovated the classrooms into laboratories. I want to tell you a little bit today about some of the things we do, but we try to do a variety of things across the field of genetics, including work with students, developing curriculum, training teachers, developing things that we can do on the internet and analyses that we can do there, and licensing the way we go about things. We really like to keep track of how many students are exposed to the laboratories that we've developed, both in our own centers here, but also at institutions around the world that use our methods or very similar methods, as well as kits that are sold and our work online. And this adds up to well over 500,000 students per year. Here at the Center in Cold Spring Harbor, we expose about 31,000 visitors per year, mainly during field trips during the academic year, instruction that goes on in the student schools, summer camps, which are going on now, and teacher training that's ongoing year-round. Some of the experiments we do are sort of old, but fun. Here's a student cutting DNA with restriction enzymes and separating them by electrophoresis. The key thing about this is that this method for working with restriction enzymes in DNA was in fact invented here at Cold Spring Harbor in the 1970s. We do more ambitious things. Beginning in 1997, we started to let students sequence some of their own DNA from the mitochondrial control region and submit that to a database. This was really the first personal genetics project that was open to mere mortals. It was well in advance of 23andMe and Ancestry DNA. And that gave students a chance to look at an electropherogram of their DNA and then can compare their DNA to other students around the world, people around the world, and even Neanderthal. Another one of our signature projects has been the development of DNA barcoding for authentic student research projects that typically extend over a half a year or a full school year. Here they are at the annual symposium that's held both here at Cold Spring Harbor and in New York City. And to give you an idea of the impact of this, the 3 different barcode projects that we run have reached about 3,000 students over the last 7 or 8 years and resulted in a number of sequences, barcode sequences published to GenBank with students and teachers as authors. Well, it's really fun, and you can easily see that everyone has a great time working with a centrifuge, learning how to do gel electrophoresis, pouring gels, working with micropipettes. And this is universal. Everyone loves these experiences to see bacteria glow when expressing GFP. And especially on Parent Day, when families come and share the experiments with their children, it's a really joyous time. That was the talk that I was going to give. But things changed, the world changed. And the DNA Learning Center shut down like many places around the world. For us, it was March 16th, 2020. And we were left asking the question that we all had to ask: what do we do now to pick up the pieces and go on? First, we had started even before the pandemic reached New York by doing virtual lab telecasts for our colleagues in mainland China, doing 2 experiments, restriction enzymes and transforming bacteria in this case. So when we had this shut down, we resolved to keep busy and to transform ourselves from an in-person lab place to a virtual lab place. And we instituted what we called DNALC Live. And over the next 2 months, DNA Learning Center staff presented the labs that we normally do from their home kitchens, such as this case. With moderated chats in the background. We gave museum tours, courses, teacher chats, short series, and we published 10 to 12 live events per week. As well as producing from the instructors' own homes, we also set up a lab as a studio and did more entailed kind of labs from this sort of makeshift studio. Over that period of time, we generated more than 100 live events. They ranged from introduction to CRISPR, to zoology, to extractions, to insects in your backyard, bacterial transformations, and so on. During this time, here in blue, we see we published all these things to YouTube, and if you compare our YouTube viewership last year in 2019 to this year in red, at the peak of the COVID outbreak, our YouTube visits had almost tripled. And even now that we've gone out of this mode of daily live labs, our viewership has still more than doubled. And we wondered how we did in relation to other institutions that do science education. And we were really proud to see that we couldn't find any other institutions that had published as many YouTube videos during this COVID period, even though many had many more visitors than us, many more had more YouTube channel views over the life of their channel, and many had more subscribers than us. Now we're in the summer season and we've shifted from in-person camps to virtual camps. And I'd like to show you what's going on this very week at the DNA Learning Center. First off, here's our schedule. We have 6 different camps going on. Each one goes about 2 hours per day. Down in one lab, the youngest students, 10 and 12, are studying cell biology and basic genetics. Here's a class on enzymology and an experiment with catalase. Here's the students working. Here's a course in forensic biology. On this day, students were analyzing their own genotypes for DNA polymorphism that they amplified in their homes using isothermal DNA amplification that does not require a PCR machine. And here, students are analyzing barcode sequences that they developed last week. And here's a course on biocoding. And in the background is another of our teachers who is actually monitoring that experiment, but at the same time setting up experiments for next week. The whole place has been converted over to a little factory. Here are Drosophila mutations that will be used to show the impact of genotype to phenotype. Here's preparing for DNA barcoding. Here's what the kits look like that we send out to each student the week before the experiments. These are 3 different kinds of kits that allow them to do some of the experiments at home. Here's an experiment that I'm involved with where I'll be training teachers from around the country. On July 16th, we mailed them out a little tiny kit to do DNA extraction, and then they send back to us by regular mail these little discs of filter paper that in this case have plant DNA that can be readily sequenced and DNA barcodes developed from in time for the experience. And we're doing really rather well this week. We have almost as many virtual visitors this week in 2020 as we did last year, although we don't expect our income will be the same because here was our registration at this point last year for the entire summer, and this year for the entire summer. It's about half. And 25% of these registrations are actually for doing on-demand labs where they can do these experiments at their own pace. And missing entirely from this summer, and tragically, are the number of students that we entertain each summer from Beijing 166 school. Well, you may ask, how are we able to sort of mount this response to COVID? And the answer is we've really been doing it for quite a long time. Back in the mid-1980s, we were the first people to pack up an entire molecular genetics lab and move it around the country in one of these shiny vector vans. And over that period of time, we've trained more than 13,000 students in all of the United States and many countries of the world. We've developed kits that are sold by Carolina Biological so that teachers can readily do many of these experiments. Here are the annual exposures to those kits, now reaching over 350,000 students. And along the way, we started to interact with lots of people around the world, sharing with them our ideas in some cases, and then modeling programs after us, but also developing an explicit licensing program where we could share directly with resources with centers that would like to emulate us. And here are some of those centers around the world. I have fond memories of all of these places. But we shouldn't confuse internationalism with racial diversity. We all have to think today about how we can level the playing field in biology for underrepresented minorities in our own countries. And in the US, that explicitly means African Americans, Hispanic Americans, and Native Americans. We started trying to do something about reaching out more to these people a long time ago, beginning about 15 years ago, when we started to target some of our training courses for high school and college faculty and hold those events at minority serving institutions. And we found that when we do that, we can radically increase the percent of teacher participants who are underrepresented minorities. You can see them here where we did specific targeting. When we don't try as hard, the number drops below 20%. And interestingly enough, in our new virtual things this summer, virtual training we've done, also good percentages of underrepresented minorities. We made a commitment to really do something for students, underrepresented students in 2008, when we teamed with the New York City Department of Education and created this single lab in a middle school in Harlem, in Manhattan. And since that time, more than 32,000 students have attended field trips there. We've granted over $500,000 in scholarships to disadvantaged students. And almost 2/3 of the students who go there are underrepresented minorities, compared to about 20% here on Long Island. And the lesson here is that if you want to do a good job helping underrepresented minorities get a leg up in science, you have to go where they live and do things expressly for them. Here are learning centers that we operate in the New York metropolitan area. Here's Manhattan here. Our dream has always been to have a dedicated center in New York City. The center that we run in Harlem is just borrowed space. And now we've teamed with the City University of New York to develop a new center in central Brooklyn in this facility here, where we hope we can share all of the great things we've developed and share with students here on Long Island with more of the students of New York City. Few things about the facility. It's 18,000 square feet. It will open in spring of 2020. We're going to focus a lot on workforce education because this is part of a national center for biotechnology education called Innovate Bio. It's going to be an easy place to reach many underrepresented minorities because the natural constituency of the CUNY school system, and especially City Tech, is about 67% underrepresented minorities and also disadvantaged students. Of the 6 teaching labs we'll have in this facility in green, 4 are dedicated to high school outreach of the sort we do here, and 2 are dedicated to undergraduate research projects for CUNY students. And there's 2 bioinformatics labs as well. The final thing I want to say is how we got to Brooklyn. It's quite fascinating. This is the first class of biologists who at that time were high school teachers as well as college professors who had come to Cold Spring Harbor in 1890 to study evolution in the natural world, which would become ecology. That institution, the forerunner of Cold Spring Harbor Laboratory, the biological lab at Cold Spring Harbor, was founded by the Brooklyn Institute of Arts and Sciences in 1890. The Brooklyn Institute went on to encompass the Brooklyn Museum, the Brooklyn Children's Museum, the Academy of Music, and the Botanic Garden. So when DNA Learning Center New York City opens at City Tech in Brooklyn, we will join our sister institutions along the axis of Flatbush Avenue, which is the main road of Brooklyn. And in that way, the DNA Learning Center will be coming home to Brooklyn. Thank you.
Thanks, Dave. That was a fantastic talk. Clearly, you and the entire DNA Learning Center are doing great things for the scientific community, especially things that are valuable right now, given the COVID pandemic. And I think it's also amazing how quickly the Learning Center was able to pivot into what you called sort of factory mode for complete online tutorials and training. So that was very impressive. My first question to you is, so we have, a lot of people, listening to these talks today, everyone from early career researchers to more senior people, but, some may be interested in going into, purely academics and academic research and bench research, and other people may have alternative careers in mind. So can you tell us a little bit about what sort of the phenotype of your team there that supports the DNA Learning Center, and what types of people you have working there and creating all of this rich content?
Well, we really have a multidisciplinary team. And a third of that team, in fact, are people who have trained to be research biologists, have PhDs, who have decided to devote themselves to education. So that's about a third of the people. Another third of the staff are people who have trained themselves essentially to be biology or science teachers. And then another third is a mixture of computer programmers and multimedia designers and evaluators. So it's a mix of people, but there's plenty of opportunities in science outreach for people for whom maybe bench work isn't the exact thing and would like to try something different.
Sure. So another question is, when life returns to quote unquote normal, when you're able to have students and trainees there on campus, are there opportunities for participants from outside of New York, outside of the area that you're in? And is there any sort of sponsorship or support for that, for trainees to come from different parts of the country or the world even?
Well, again, our model has been that it's always instruction to the people. So we have the capability to take our instruction really anywhere in the world, and we do that routinely. So the first answer is sort of antithetical to what you're saying, but the best way to reach lots of people is to in fact take expertise you have and offer the exact same experience that we can offer at Cold Spring Harbor in Beijing or Suzhou or Vienna, Austria, or wherever. Now that being said, about 25% of the students who are signing up for our online course now are doing them from afar and sometimes on demand, meaning that they just take the course at the. So we actually see, we actually asked the question of a group of about 16 teachers who were doing meta barcoding training last week. We asked them whether they, in the future, whether they would prefer to have all in-person training, virtual training, or a combination of virtual and in-person training. And you know, out of those 16 teachers, not one said that they would prefer all in-person training. So in fact, we're seeing a shift in culture in all different ways now, where we're seeing that we can do a lot of things efficiently on the internet. And the hybrid training, such as we're doing now in the summer, I think is going to be very important for all kinds of academic institutions to do in order to survive, which is to do a combination of online and hands-on learning.
Thanks, Dave. Let's go to some of our audience questions. We have one here from a participant, Sarvanaz Sarabipur. Sarvanaz, are you there? Would you like to ask your question?
Yes, sure. Great talk. I was wondering if you have plans for other types of training that's going to be later important for researchers. So this is training on how to manage data, how to collaborate well on research ethics, and also providing mentorship on how to be a researcher and how to work in science.
Yes, as a matter of fact, I didn't have the slide, but I'm personally, and many of us here at the Learning Center are very interested in biological infrastructure, which is what do people need to do experiments easily and then to analyze the DNA online, the data online easily. Sorry, I lost my space. I'll get back to it. I became completely lost there. Could you ask that question, Mom?
Yes.
My mind is on a mile a minute.
It was about research culture, and
yeah.
that is not just doing science, but also using the infrastructure, as you said, and also valuing other aspects of research, for example, collaborations, ethics, and data management for reproducibility.
Right. So, I mean, what we really try to do at the DNA Learning Center is broaden the interface between research and education, where people can walk in both worlds and cross that line easily. One way we can, because this is really the first time in the history of biology that a student or teacher at a small college or two-year college or high school can work with the same data at the same time using the same tools as high-level researchers at these sister institutions. So what we try to do is to simplify the biochemistry so that you can get data, such as a data set of metabarcode data, so that you can generate a sample of metabarcode data for $10 rather than $60, which is what it costs now. And that then you can take those biological big data, such as metabarcode data or RNA sequence data, and analyze it using the same workflows and tools as working biologists do, but do it in a simple way using a graphical user interface that doesn't require programming skills. And in fact, we find that about 25% of the people who use our simplified bioinformatics tool called DNA Subway are professors, and 45% of them are actually using that tool for their own research, because it uses research grade tools, but it's a little easier to use. So my point is, if we want to develop an agile, culture of science people, then we should encourage researchers to move a little bit down towards education, and education to move a little bit towards research. And in fact, you know, this is what we find happening all the time when we work with teachers at all levels and biology faculty at all levels. Oh, the final thing is we did a survey of several hundred NSF principal investigators who held grants in the biological division of NSF. And we asked them, you know, what do you need to deal with data in the future? First off, about 90% of them said they were currently or would soon be working with biological big data set. And this spans all the disciplines of biology. Secondly, when we said, what are your biggest unmet needs? What are your biggest unmet needs? They didn't say that it was infrastructure. As a matter of fact, that was lowest on the list. There's plenty of computers, there's plenty of computer capacity, high performance. But what they said they needed most at the top of their list was training, especially in data integration, taking data from disparate sources and melding them together, and also in cloud computing. So the point of it is that even high-level biologists who win grants from NSF are falling behind in the battle to analyze their data. And so we need a lot more training.
Thank you, David. I think that is all the time we have for questions now. I just want to take one last minute and thank you both David and Michaela for great talks today. I think we learned a ton about various aspects of research and training that go on at Cold Spring Harbor. So thank you so much for taking the time to tell us about that today. I also want to thank the audience. We had nearly 400 people joining us today, and I'm sure we had many more on some secondary streams that have been set up around the world. I want to encourage everybody, especially if you didn't have your questions answered, please head over to the Slack channel. The speakers have promised me that they will go there and engage with you and any unanswered questions. The link is in the webinar chat box. And lastly, I want to encourage you to come back and join us next week on Wednesday, July 15th, same time, same Zoom link. And we will be hearing from the University of Buenos Aires and CONICET in Argentina. And the speakers will be Alberto Kornblit and Andrea Gamarnik. So hopefully we will see you all there. And thanks so much for joining us today.